Yeah, go ahead. Excellent. Okay, well, Melvin, thank you so much for that wonderful introduction. I'm super excited to be talking to the Information Physics Institute, this amazing international, interdisciplinary organization focused on information theory. And I'm thrilled at the opportunity to be able to share with you a little bit about the science that I do, the questions that I like thinking about, and also a little bit about myself as a person. So let me start off by introducing myself a little bit. Again, Melvin gave such a wonderful and thorough introduction. My background is basically in planetary science. So I'm a planetary scientist by training with expertise in atmospheric chemistry. Basically, I use numerical models to simulate the physics and the chemistry of planetary atmospheres, both within and beyond our solar system. My academic interests lie mostly in the realm of astrobiology, so I'm very interested in questions about how does life emerge, what makes a habitable planet, and how should we best go about trying to look for life elsewhere in the universe. These days, I approach these problems mainly through the lens of data science and informatics, and you'll see many examples of that throughout the course of this talk. And at the end of the talk, you'll start to see how philosophy sneaks into my work as well. One grand goal of mine is to help contribute to developing a theory of life, which of course is a grand endeavor. It may take a lifetime or maybe even several lifetimes to accomplish. But I think similar to the way that we have a theory of gravity that helps us interpret astrophysical phenomena, and how we have a theory of chemical kinetics that helps us interpret astrochemical phenomena, I believe that the field of astrobiology will greatly benefit from a theory of life, and I have a hunch that that theory of life will have to do with information. And so today, obviously, we're not going to go so far as to develop that full theory of life, but hopefully you'll see how we are inching closer in that direction, taking positive steps forward. So I titled this talk An Informational Perspective of Life because I really think that information processing is at the heart of what life is and does. And so let me tell you about a paper that I wrote with my colleague Anirudh Prabhu last year. So Anirudh is a data scientist by training. He's an expert in machine learning and artificial intelligence and the application of such methods to all kinds of Earth and planetary science problems. And for 2 years, we shared an office here at the Carnegie Institution for Science, and our frequent interactions have led to many exciting projects, fun ideas at the intersection of data science and astrobiology. And so in this paper that we published last year, titled Cells as the First Data Scientists, we're basically arguing that we see the activities of data scientists as the latest development in life's ongoing journey to better understand and predict its environment. So prior to joining Carnegie, I was really a novice at data science. I had never taken a course in data science. I barely knew how it worked. So I learned a lot from Anirudh. And Anirudh showed me something that was called the information life cycle in data science, which describes the way that data scientists collect, treat, and organize and refine information in an autocatalytic fashion. So the first step of any data science endeavor is obviously the acquisition of data. So you could collect data through observation or through experimentation, or even rescuing it from scattered resources like old PDFs of papers. The second step in data science is to curate that data, to clean it, format it, and make it useful. The third step is preservation, meaning retaining that data in usable form so that it can be accessed in the future, both for the original purpose or for purposes that were not even imagined at the time that it was acquired. The fourth step in this information life cycle is stewardship, which means maintaining the integrity across acquisition, curation, and preservation. And finally, we have management, which is creating the infrastructure that oversees all of the other processes and facilitates further data collection. Now, as somebody who was very interested in information processing within living systems at large, I saw some clear analogies to the way that life processes information. So in life acquires new data, new strands of information through various tweaks to its genetic code. And that information is then curated through natural selection, which prunes a wide range of information-bearing states to a smaller number of viable ones. Preservation is achieved through replication, reproduction, and growth, and stewardship is performed by error-correcting mechanisms. The management stage involves mechanisms that control gene expression, like epigenetic markers, gene regulatory networks, and factors that tune mutation rate and the uptake of new genes from the environment. We can also take this analogy one step further by seeing how once stewardship and management evolved, they would have cemented themselves into the information life cycle through benefiting, preservation, evolvability, and the acquisition of new data. And this is very similar to the feedback that occurs in data science, where once you have stewardship and management, you facilitate new data sharing amongst different parties at different institutes and universities. You facilitate collaboration. You help acquire new data as well and process it. Okay, so that's one thing. The other thing that Anirudh taught me early on in our collaborations was about this idea called the data information knowledge ecosystem in informatics, which describes how data, information, and knowledge are all represented and communicated meaningfully in informatics. So raw data, they're created and gathered, but they're generally useless to the vast majority of people. So informatics methods are required to transform that raw data into information. So information here means data that is represented and can be understood by consumers, whether those consumers are scientists or even just the general public. In other words, information is data that is represented and organized in a meaningful context. Once consumers are armed with the appropriate information, they are able to integrate that information with other experiences to produce what we call knowledge. Knowledge, therefore, is not something that is acquired in solitude. It's a collective phenomenon that is reliant on experiences and context. And so as a whole, the field of informatics views itself as the body of work that seeks to transform raw data into information and information into knowledge. So one last remark here, you see those arrows here connecting data, information, and knowledge are bidirectional. And that's because once you have, say, data transformed into information, like you imagine data collected from the field, transformed into information in scientific papers, that information can become data for some other future endeavor. So for instance, somebody might do a meta-scientific analysis, connecting a bunch of research papers together using a co-citation network or something like that. Okay, so once I saw this, I was like, oh, wow, this is remarkably similar to what happens in living systems as well. So for biological organisms, data will represent the physical features of the environment, the fitness landscapes, the environmental parameters. You've got your pH, your seasonal cycles, your temperature, salinity, what have you. And by sensing and computing their environment, living organisms actually transform that data into information that is relevant to their survival. Now, an individual biological unit of selection is basically a proposal from the biosphere to the environment. It's like a prediction that is put to test against new data. And so via natural selection over multi-generational time, certain predictions will be strengthened while others will be discarded. And populations of individuals that we call species will gain lasting quote unquote evolutionary knowledge about their environment. These are recipes of success that are written in genetic code. And hence knowledge in the biological sense is very much like knowledge in the data science sense. It is gained through experience over time at the communal level. Okay, so let's recap what we've learned so far. We've seen how several concepts developed to understand and explain the activities of data science and informatics are strikingly descriptive of the way that life in general processes information about its environment. This includes the idea of the information life cycle, as well as the data information knowledge ecosystem. In other words, the activities of data scientists are just the latest development in life's ongoing journey to better understand and predict its environment. The principles of data science we see actually expressed across the expanse of biology, from microscopic viruses to the largest clonal tree to your friendly AI engineer. Okay, so with this fun warm-up exercise out of the way, let's now turn to some of our work on at the intersection of astrobiology and data science. So as we've seen, informatics and data science are powerful lenses with which to view the living state. And this is because life is the quintessential complex evolving system. And life's inherent complexity, this complexity both at the molecular and the planetary scale, demands that we take multidimensional approaches to trying to understand it. Now, one way that life impacts its environment is through atmospheric chemistry. So let's now talk about atmospheric chemical networks as agnostic signatures for extraterrestrial life. This is work that I led in collaboration again with my colleague Anirudh Prabhu, as well as Jason Williams, Shauna Morrison, and Bob Hazen at Carnegie Science. So the dream is one day to be able to take an exoplanet target, a planet orbiting some other star, and examine its atmospheric chemical makeup through the various modes of spectroscopy, and determine using those data whether or not this planet comes from the subset of planets with life or the subset of planets that are barren. And in this talk, I want to make the case that a high-dimensional phase space of network metrics might be one of the best ways of trying to do this. So what do I mean by network metrics? Well, the chemistry of a planet's atmosphere can be viewed as a graph, where the nodes of the graph are the different molecules or chemical species in that network and in that atmosphere, and the edges of the network represent the reactions among those species. So by way of example, this is phosphine in a subset of Saturn's atmospheric chemical network, phosphine pH3. And we see in this network that it is directly connected to 5 other chemicals. So in network science parlance, we would say that this particular node here has a degree of 5. Here I'm showing you the network diagrams of atmospheric chemistry in 4 different solar system worlds. So we have Saturn's moon Titan, our sister planet Venus, a model of early Earth and early Mars, and our modern Earth right now. In these networks, the nodes are colored by their degree. So again, that's the number of other nodes that node is connected to. And they are sized by their betweenness centrality, which is a measure of the number of shortest path lengths between other nodes that passes through that node in question. So hopefully you can see just by eye that these networks have very different topologies to one another. And so, for instance, Titan's network is very symmetric and densely clustered around hydrogen chemistry at the middle, whereas Venus's network is pulled into a kind of lopsided shape by the various oxygen, sulfur, and chlorine-bearing radicals in its atmosphere. Early Earth and modern Earth have their own unique hub nodes. For instance, in the more chemically reduced early Earth atmosphere, HCN would have played a pronounced role, whereas on modern Earth, it's more about nitrogen oxides and even the chlorine-bearing compounds that we've pumped into the atmosphere. So you can already glean so much just by staring at these networks with your own eyes, but you can also quantify how they are different to and similar to one another. And that's what I'm doing here in these 2 global network metrics. On the left, in this left bar graph, you see the network degree assortativity. This is a measure of how heterogeneous the network is. So the lower the number, the more heterogeneous the network. And on the right bar graph, you have the transitivity. This is a clustering coefficient. Basically, it's asking for any given node in the network, are the neighbors of that node likely to be neighbors of each other as well? And so what you can see here is that Earth's network topology not only differs quite markedly from those of other planetary atmospheres, but is actually more similar to the network topologies of living systems. So in the various shades of green, I'm showing you a metabolic network, a neural network, and a marine food web network. So there might be something about life, the way that it organizes matter and information and energy flows at a way that unifies all these various disparate scales from the subcellular all the way to the planetary. And the big question that we wanted to ask about the atmospheric networks is, is this a result of the fact that Earth's atmosphere has co-evolved with our biosphere for 4 billion years, and more recently our technosphere? And that's a big question. We're not all the way done answering it yet. But the first step to getting there is to just ask ourselves, in what other ways is Earth's network distinct from those of other planetary bodies? So one common way of characterizing complex networks is by so-called centrality metrics. Now, centrality metric measures the importance of any given node to the network, but importance is a very vague and ambiguous concept. There are many different ways of being important, and hence there are many different centrality metrics at our disposal. The classic ones are degree, closeness, and betweenness centrality, where degree centrality equates importance to the popularity of a node, closeness to a distance metric. Betweenness is a measure of control that node has over the connectivity of disparate parts of the network. But I think all of these classic centrality metrics are imperfect for our atmospheric studies. Degree centrality is a very local metric. It doesn't really care about what's going on beyond a node's nearest neighbors, whereas closeness and betweenness centrality make this assumption that influence can only travel along shortest paths through a network, but that may not be so. And so I like to turn to some of the more esoteric centrality metrics, one of which is information centrality. So in information centrality, we allow influence to pass between two nodes, not just between their shortest paths, but between all possible paths in the network between those nodes. So this centrality metric appeals to me for characterizing our atmospheric networks because we know in atmospheric chemistry that chemicals don't just influence each other directly. For instance, there are many catalytic cycles that provide indirect routes for precursor molecules to create and form their end products. So in this graph here, what you're looking at in the violin plots are the distribution of information centrality in all of the different planetary atmospheric networks that we're studying. And you can see from this plot that the distribution of information centrality in modern Earth's network is very different from most of the other planetary networks, except for maybe Jupiter. And this is actually a trend that we've started to notice that in certain network metrics, Earth and Jupiter's networks look very similar. Our hypothesis is this is because Jupiter's atmosphere is very different from all these other planetary atmospheres. It doesn't have a solid surface, which means that it can dredge up through convection all of these out of equilibrium chemicals from the deeper recesses of the atmosphere, adding to the disequilibrium, to the complexity of the chemistry that we observe in the photochemical region. Now, given that, is there a way to distinguish between Earth's network and Jupiter's network? Indeed, there is. If you move into a higher dimensional phase space, so in this three-dimensional plot, I'm showing you information centrality on the z-axis, that's what we were looking at before in those violin plots, and then two other centrality metrics on the x and y-axis. We've got the average neighbor degree and the log of the subgraph centrality, which is yet another centrality metric that deals with information flow. And so in this 3D plot, you see how the various atmospheric networks start to segregate from one another and occupy their own unique regions of this phase space. This is just one of many possible n-dimensional phase spaces that we might want to explore to be able to distinguish atmospheric networks from one another. Another way that we are characterizing these atmospheric networks is through community detection. So we've run what's called the Louvain community detection algorithm. This is an unsupervised machine learning algorithm that optimizes the modularity of these graphs by assigning each node to a distinct community. And what we found is that Earth's network typically produces more different communities than the other planetary networks. Another thing that we're doing is hierarchical clustering. So these heat maps show you the distances between different chemicals in the atmospheres, where black is low distance and yellow is high distance. So the diagonal black line that you see is basically the zero distance between one chemical and itself, right? So there's zero difference between, say, oxygen in Earth's atmosphere, but there might be some difference between oxygen and methane in Earth's atmosphere. And when you create a heat map like this, you can see various structure and complexity emerging in these diagrams, and you can start to assign that structure to different chemicals. So on Jupiter, for instance, a lot of this complexity arises from phosphorus chemistry. Indeed, if you took out the phosphorus chemistry from Jupiter's atmosphere, you'd have a relatively bland hydrocarbon nitrile atmosphere like that of Titan. Venus's atmosphere gains some of its structure from the sulfur molecules that are produced in the atmosphere. And Earth's atmosphere gains some of this structure from the chlorofluorocarbons that we've pumped into the atmosphere hence making this type of analysis applicable not just for biosignature research, but perhaps for technosignature research as well. One last crucial step that we wanted to test was to compare our real-world networks to equivalent random networks. So basically, an equivalent random network is taking our real-world network and scrambling it. So it has the exact same number of nodes and edges as a real-world network, but it's generated completely randomly. So on the left here, you see modern Earth's network topology. You have the equivalent random network to modern Earth's atmosphere on the right. And so you can see how much more homogeneous it is. Clearly, Earth's chemical network is not just random chemistry. Indeed, Earth's network is actually the most non-random of all of the planetary networks. And if you ratio the various network metrics that we've been discussing between their values for the real-world network and their values for the equivalent random network, Earth's again stands out, whether you're talking about transitivity or subgraph centrality or something else. Okay, so what have we learned so far about atmospheric network topology? Number one, Earth's atmospheric network is distinct from those of other planetary bodies in numerous ways, and in certain metrics, Earth's network resembles those of living networks. And this is not simply due to the fact that Earth has a unique number of nodes and edges, in other words, that we know more about Earth's network than other planetary bodies, because compared to their equivalent random networks, Earth still stands out. And so something that I want to be able to try to develop using network science, graph theory, and other information theoretical metrics is over the next 5-10 years, hopefully try to understand whether network science can reveal a general class of agnostic biosignatures at the planetary scale. And I really believe that now is the time to do this crucial hard theoretical work, because as an astronomical and planetary science community, we get once per generation opportunities to build facilities that will revolutionize our search for exoplanetary life. And we want to be able to inform our engineering and instrumentation colleagues about what detection capabilities we need to actually distinguish whether a planet has a biosphere versus a planet that does not. But even more time sensitive for me is the development of in situ biosignature techniques for solar system exploration. And why is this? Because we are embarking on a golden age of astrobiology in the solar system, where numerous spacecraft are being developed and launched to solar system bodies with the explicit goal of trying to detect signs of habitability and life. So for instance, we are in the saga of a Mars sample return mission, trying to get samples from the red planet back to Earth. We're launching a mission to Jupiter's moon Europa called Europa Clipper later this year in mid-October. I hope we're all, watching that as it launches and goes off. Dragonfly, this octocopter mission, is going to visit Saturn's moon Titan in the 2030s. And the latest NASA decadal survey, planetary science decadal survey, recommended a flagship level mission to visit Saturn's moon Enceladus to search for life in its icy plumes. So we're really embarking on what could be a golden age of astrobiology. And so now is the time to think creatively about how to look for life. Four necessities for a strong biosignature technique, in my mind, there are four. Number one is a robustness. So this means as immune as possible to false positives and false negatives for detecting life. Number two, the technique should be agnostic, which means capable of identifying life even if it doesn't share the biochemistry of life on Earth. Three, that it would be probabilistic, which means that we can quantify our confidence in the detection of alien life or our non-detection of alien life, whatever the case may be. And finally, that it is flight ready, which means that it relies on instrumentation that NASA, ESA, or some other space agency is actually going to strap onto the back of a rocket and launch into space. If it doesn't, then it's probably not going to be used in the near future. So the biosignature technique that me and my colleagues at Carnegie have been developing in the past few months, a couple of years actually, is one that seeks to accomplish all four of those main criteria. So this is a robust agnostic biosignature based on machine learning and pyrolysis GCMS. This technique was published last September in PNAS in a paper that was led by my colleague Jim Cleaves. So say you want to find life on Jupiter's moon Europa, what are you going to do? You're probably going to send a spacecraft to sample some of this surface material, this plume detritus that probably originated in that habitable subsurface ocean below. You might fly one of these instruments called a pyrolysis gas chromatograph mass spectrometer. Why? Because these instruments are flown all over the solar system regularly. In fact, as we're speaking, there is one sitting on the surface of Mars. So basically, if you're not familiar with what a pyrolysis GCMS is, it's just an oven that not only bakes your sample, but tastes it for you too. This is the kind of data that a pyrolysis GCMS gather. On the x-axis, we have the mass-to-charge ratio of different ions that are being counted in the instrument as the GCMS heats up in temperature over time. So the z-axis is simply the intensity of those signals, the number of molecules per second of that mass-to-charge ratio at that temperature that you are counting. Now, if you found something that looked like this on Europa, I think you'd be very excited because this is what the average Earth bacterium looks like. Thing is about looking for life on other planets, other worlds, is that there's no guarantee, again, that they will use the exact same biochemistry as life does on Earth. So it would be kind of foolish to design a life detection experiment by looking for any particular molecule, like one of those particular peaks that you see there. Instead, maybe there's something about the overall distribution of molecules in a sample that tells you whether it comes from something that was alive or something that was not alive. And so what we've been doing at Carnegie is running a bunch of diverse organic rich samples through our GCMS to gather a whole bunch of data, samples that were once alive very recently, like bacteria and plant matter, things that were alive a long time ago and over millions of years of geologic processing have turned into petroleum or coal or shale, and organic rich samples that were never alive, such as carbonaceous chondrites, a class of meteorites that probably contributed to the building blocks of terrestrial planets. And hopefully, again, just like those atmospheric networks that I showed you a few minutes ago, just by eye, by staring at these four different kinds of samples, you can see that the distribution of organic molecules in each of them is different from the others. And so when we saw this, we're like, wow, this is so amazing. Maybe we can quantify how they are similar to and different from one another, and also train a machine learning algorithm to try to tell the difference between something that was once alive and something that was not. And so that's exactly what we did. We trained a random forest algorithm on an initial sample set of 134 diverse organic rich samples. And the final algorithm was able to tell the difference between something that was alive and something that was never alive to 90% accuracy, which is a huge leap for astrobiology. Now, what's really cool is that the algorithm could also tell the difference between something that was degraded biotic from fresh biotic. So the difference between like a coal versus a fresh bacterium. And that's really cool because we didn't actually tell the algorithm how to do this, right? We only taught it the difference between something that was once alive and something that was never alive. So what you're looking at here is a graph where we're looking at the first two principal components of the data, the first two of 20 principal components. And the abiotic samples cluster in a very tight region of this parameter space, with the biotic fresh things sweeping out in green to the lower right, and the biotic degraded things sweeping out up to in the blue. Now, you might be concerned about the overlap that you see here, but again, remember that these are just the first two of 20 principal components. So if you imagine spreading this graph into a third, fourth, fifth dimension, etc., the clouds of points will further segregate, hence the 90% accuracy of this algorithm. Now, as an astrobiologist, what's really exciting for me is the potential to use this to find what we would call biological anomalies on other worlds. So imagine if you went to Europa or Enceladus, you sampled some organic matter, and you found that it plotted in this diagram in one of these question mark regions. That means that you found a sample whose distribution of organics is markedly different from not only all known abiotic samples, but also from the distribution of organic molecules in life as we know it here on Earth. And I think if we found something like this, it would be a tantalizing biological anomaly that could lead us to the discovery, potentially, of life as we don't know it, life with a very different biochemistry. So we've got two great opportunities ahead of us. Obviously, in astrobiology, we have now this robust agnostic biosignature technique that has the potential to identify life, even life with non-earthly biochemistry. And then on the other hand, we also have this amazing opportunity in paleobiology, because in the paleobiology community, different groups have been at each other's throats for the past several decades over the idea that certain 3 to 4 billion year old ancient rocks may have traces of the earliest life forms on Earth. Some people say, yes, those rocks have microfossils. Other people are like, no way. Well, maybe our machine learning technique will be able to provide an independent line of evidence that can help us understand better the evolution of early life on our very own planet. Okay, so why fundamentally should it be the case that the distribution of organic molecules in living samples should be different from the distribution of organic molecules in non-living samples? Well, here's where I want to make an analogy to statistical mechanics, right? So imagine you've got a hot cup of tea, sitting on your desk, right? So the microstate of that hot cup of tea is going to be given by the position and the momenta of the particles that make up that teacup. And there are vastly more microstates that correspond to a cold cup of tea in a room temperature room than correspond to a hot cup of tea in a room temperature room. And so this is why our cups of tea tend to cool down with time. This is our second law of thermodynamics. This is the arrow of time. Now, the analogy that I want to make to life is this. There may be a similar parameter space, not of position and momenta of particles, but a high-dimensional phase space of molecular patterns that helps us distinguish between living matter and non-living matter. Now, the curious thing about life is that it seems to evolve in a way that is opposite to the arrow of thermodynamics. Non-living worlds become living worlds, and we have origins of life, and biospheres evolve and spread. This seems to be in defiance of the second law, but of course, the key is that life is an open system. It's able to trade information and matter and energy with its environment. And it also responds to selection pressures, namely selection for function. So we explored this idea of selection for function in a paper that I led titled On the Roles of Function and Selection in Evolving Systems. And this is a paper that I'm very proud of because it represents intense discussions for a period of over 2 years between not just an interdisciplinary group of scientists, but also between scientists and philosophers of science. So for example, Professor Carol Cleland and Professor Heather Demarest, these are philosophers of science at CU Boulder in the United States, who've really sharpened the ideas in this manuscript. And I'm really grateful for their contributions to our scientific endeavor as philosophers. So the goal of the study was to try to introduce an explanatory framework for the evolution of physical systems, right? So the second laws of thermodynamics, and as this group well knows, infodynamics, are the only time asymmetric laws so far articulated in the scientific canon. But these laws do not explicitly address the tendency for evolving systems to become increasingly complex with time, and therefore a missing law of cosmic complexification must be consistent with all of these previous laws, thermodynamics, infodynamics, gravity, electromagnetism, etc., but may not inevitably follow from them. So we began our investigation by noting that natural laws are built around conceptual equivalencies. For instance, Newton's law of gravity was built upon this esoteric property of having mass that describes myriad disparate systems, whether it's an apple falling from a tree, a planet orbiting a star, even atoms and molecules floating in planetary atmospheres can be described by or influenced by Newton's law of gravity by virtue of their having mass. So the first question that we wanted to ask ourselves was, what conceptual equivalencies underlie all evolving systems? So for us, evolution occurs in any systems that have three properties. Number one, a large number of interacting components, diverse interacting components. Number two, a way to sample myriad configurations, explore configuration space of those interacting components. And three, selection for function. Let me give you a few examples to illustrate what I'm talking about. So in nucleosynthesis, our interacting components are protons and neutrons, and the ways that we sample new configurations of these protons and neutrons are through stellar fusion, supernovae, stellar mergers, and there's a selection for stability. In mineral evolution, our interacting components are at one level higher. They are the elements of the periodic table. And you sample new configurations of these elements through the various modes of mineral paragenesis. As planets evolve, they subject their materials to new pressure, temperature, composition spaces. And again, there's a selection for stability. And finally, in biological evolution, our interacting components are many and at many scales. We've got genes, cells, species, and you sample new configurations of them through mutations, horizontal gene transfer, recombination events, etc. And we have a selection for survival and propagation in biology. Now, the most mysterious thing to me at this juncture was, what do we actually mean by selection for function? In other words, what are the most fundamental selective forces in nature? What functions does the universe, quote unquote, care about? So in our paper, we seek to identify the three fundamental sources of selection in nature. So the first is static persistence. This simply speaks to the idea that configurations of matter tend to persist in time unless kinetically favorable avenues exist for their incorporation into more stable configurations. Now, static persistence allows there to be out of equilibrium structures in the universe whose dissipation can drive emergent dynamic systems, such as convection cells and wildfire and even life. And within these dynamic systems, functions may arise, where functions are defined as any process that has causal efficacy over that system's dynamic persistence. And finally, we think that there is a selection pressure for novelty generation because the discovery of new functions that promotes dynamic persistence raises that system's quote unquote kinetic barrier against decay to equilibrium. And we see novelty generation all over the living world, from the acceptation of one function to another, or the way that bacteria can tune their mutation rates such that they mutate faster in more stressful environments. And so finally, given this functional view of evolving systems, what might be an auspicious ontology for a law of increasing complexity? Here's where we turn to this idea of functional information, which quantifies the fraction of all possible configurations of a system that possesses a certain degree of function. Functional information was first introduced by Jack Szostak in 2003, further explored and refined by Bob Hazen in 2007. And it basically speaks to the fraction of possible configurations of a system that can perform a certain degree of function. It is defined simply as the negative log base 2 of that fraction of configuration space that can perform that function. So given this definition of functional information, we have to ask ourselves, for any given evolving system, why would the functional information of that system change with time? Well, there are two fundamental reasons why it could change with time. First is you might have strengthening selection pressures. If you confine the hypervolume of configuration space that can perform that function to a smaller region, then you have decreased the fraction of states that can perform the function and increased the functional information. On the other hand, you might have a configuration space expansion through say, the addition of components to the system. And again, if you increase that configuration space, you decrease the volume of configurations that can perform the function, and you've increased the functional information. Let me give you a concrete example of functional information increasing in the non-living world. So this example comes from the vast literature on mineral evolution. In mineral evolution, you have configuration space expansion because of the ever-increasing diversity and availability of elemental components that can comprise minerals, as well as the idea that planetary environments will sample new regions of pressure, temperature, and composition space as time progresses. And you also have strengthening selection pressures due to the ever-increasing time between crystallization of any given mineral and the present, and the fact that new minerals arise by modifying and changing the prior generation of minerals. So we've actually quantified this in a new paper that we have submitted and gotten accepted in PNAS Nexus. It should come out anytime now, potentially as soon as Monday of next week, where we can't quantify the functional information of Earth's mineralogy through deep time. So we start with the first minerals that were ever created in the universe in stellar atmospheres, through the minerals that would have been created in the protoplanetary disk, through the minerals that we find in meteorites, which are representative of the asteroids that built planets, through the minerals that were created in Earth's earliest Hadean crust, the minerals that were created as a result of igneous and metamorphic processes resultant from plate tectonics, all the way through the biotic Earth, where many minerals are created through biomineralization in a co-evolution with our biosphere. And you can see this monotonic increase in the amount of functional information in mineralogy. So, basically, to conclude this portion of the talk, we are proposing a new natural law, emphasis on the word proposing, right? It needs to be tested. Minerals provide one test, but it needs to be tested in a wide range of living and non-living systems. And this proposal is called the law of increasing functional information. The functional information of a system will increase, that is, the system will evolve, if many different configurations of the system are subjected to selection for one or more functions. Now, what are the implications of a law like this? Well, there are many. I'll just touch upon a couple. The first is that Darwinian evolution, we can see it as just a special case of a much more general phenomenon of evolution writ large that applies to all kinds of physical and chemical systems, including mineral evolution. And also, to tie this back to some of the quantitative work from earlier in the talk, there may be a distinction between life and non-life that is related to selection for function. So the distribution of organic molecules in life have been sculpted by selection for myriad biochemical functions, whereas the distribution of organic molecules in, say, meteorites has been sculpted simply by selection for static persistence. Okay, so to wrap up this talk, I titled it An Informational Perspective of Life, because information processing is at the core of what life is and does. And information theory, I hope, will help us answer age-old questions such as, are we alone in the universe? And where do we come from? And what even is life? So today, I hope you've seen how the principles of data science are expressed across the expanse of biology, and data science is just the latest development in life's ongoing journey to better predict and understand its environment. I hope you've been intrigued by how Earth's atmospheric network is distinct from those of other planetary bodies in numerous ways, inviting the development of a planetary scale biosignature relevant to exoplanet research. I hope you've been excited by this novel biosignature technique that combines pyrolysis GCMS measurements with machine learning to achieve 90% accuracy in the differentiation between biotic and abiotic samples. And finally, I hope your mind has been tickled, if not expanded a little bit, by this proposal of ours for a new natural law based upon functional information. And with that, I'll end my talk and be happy to take any questions that you have. Thank you very much.